All right, this is Mr. Labonte. I'm going to be talking about cancer. No cartoon here because it's not really appropriate. So, first of all, let's look at aging. And so I'll talk about what's going on top to begin with. But so as you age, right, there are certain factors that are going to affect the cells. So one of them, the one that we've talked about, is as cells differentiate, you get these epigenetic changes. These are changes outside of genes, but basically certain genes are locked down. And so you're going to have changes where the genes are locked down, they're not being expressed. We've talked about oxygen as being a poison. And so as you age, your mitochondria become damaged. This is both true in mice and humans. I'm just up here for some reason. The cytoplasm becomes degraded. Extracytoplasmic matrix, cytocellular matrix is ECM. And DNA itself becomes damaged. We'll talk a little bit about that in this presentation. And then hopefully we'll be able to talk about telomerase erosion because it's important. But in all cases, this is why we age. When we do that, we get what's called senescence, or we get cells that stopped reproducing, and then that makes us look like this. This is, in fact, collagen is the type of cell that stops reproducing, which gives you wrinkles. And so this is the natural thing that happens in all bodies. And so the message, when you get damaged DNA, or when you get shortened telomerases, or when you get mitochondria that are not working, to the cell is, hey, you should not be reproducing. Additionally, when you get crowding, so we'll just pretend there's too many cells. Here are lots of cells, CR. So when you get crowding of cells, the cells get a message, hey, you should not be reproducing. So when you get cancer, there's two things that happen. The signals that tell the cell to start reproducing aren't there, but the cell reproduces anyway. And there are signals to stop reproducing. There are signals saying don't reproduce, and the cell ignores those. So those are the two things that really are the kind of, if you want to break it down conceptually, the cause of cancer. All right. Where are these signals and how do we see them? The reason that we spent so much time on the cell cycle is because the signals can be seen as being here. Since we started, really I think the conceptual beginning is G1. You go over here and you look right here before you go through synthesis. So the cell is asking, should I replicate my DNA? Okay. Is the cell large enough? Does it have enough stuff? And then other questions are, what are the signals from other cells? And what is the status of the DNA? So these are two questions that the cell should be asking itself at this point. When it gets over here, these are replication questions. This is, has the DNA fully replicated and have the spindle fibers attached to the centromeres, and are the chromosomes lined up? These are these other two questions. But right here, generally, this is the important point to have questions. There's opportunities to stop this process at other points, but this is where we're looking right here at this first stop sign. All right, so this is a mathematical way of looking at cancer. As a preface, know that we have millions of cells being created in our body and millions of cells dying. For an adult, that should mean that our birth and our death rate are about the same, not for you all. And that cancer is really just when any cell in your body is either dividing too quickly or is not dying when it should be dying. That's what cancer is. So since you have tons of different types of tissues, there are hundreds of different types of cancer because this can happen to any cell. So this first number here is 0.5 times 10 to the negative 9th. This is a chance of DNA mutation. So for any given cell, you have a 0.5 times 10 to the negative ninth chance that there's going to be a mutation in that cell. However, we have 37.2 trillion cells in our body. That's 3.7 times 10 to the power of 13. This is much larger than this number. Just as a simple mathematical consequence, what that means is there are about 37,000 cells in your body that have mutations to their DNA, right? So these are cells carrying mutations to their DNA. If those mutations do one thing, cause growth, absence, growth indications, or 2, refusal to die, all right, no die, that's going to cause cancer. So if we live long enough, we will all get cancer. That's a given. So that's the mathematical component of it. But it's a little more specific than that. Cells that divide more often, okay? So this is stem cell divisions. And so this is, I think the number down here is the number of cells that are created by this. So all the way over here, you're looking at right, these are very, this is the inside of basically your intestine, right? Your skin cells, so you have a lot of skin cells here. And then, let's see, thyroid, this would be a germline cell, so that's a germ cell. And so as you're going along, these are cells that are dividing, and then on this side, you're getting risk of cancer across your life, right? Now, you're like, wait, that's the total number of cells in your body. How can you have a cell that divides the same as the total number of cells in your body? Well, you have a lot of death, right, as well. So if you think of skin cells, right, they're dividing a ton, but they're also sloughing off and dying. So that's how you can get those numbers. But what you can see here, I think really clearly, is that the more cells divide, the more likely they are to get cancer. That is not the only factor that relates to cancer. For instance, you see that line right there, right? External factors also are going to influence cancer. But this is a general principle. Okay, so you've heard, now I'm going to go a little bit into some of the terminology that you probably heard of with cancer. So you probably have heard of these terms benign and malignant, okay? And what I want you to understand here, we'll talk about this a fair amount. I've said this a couple times, that we're sacs inside of sacs inside of sacs, right? So benign tumors have a well-defined parameter, and what that means is that they do not go outside of whatever protective tissue sac that they're in. Malignant tumors will basically invade and move through the specific tissue sac that they're in. That means not localized, okay? Benign tumors can still be dangerous. They'll press on blood vessels or nerves. You can have issues that way. But malignant tumors are much more dangerous because they will start invading neighboring tissue. Cells all become cancerous gradually. That you don't have a cell that's like, let's call the cell, whatever, let's see if I can find a cell here. That cell right there is Bob. All of a sudden, Bob doesn't become cancerous. So it has a couple of traits that when you look at them, make them appear cancerous. So as they're replicating, and even in the life of the cell, they get enlarged nucleus. The edges, basically the nucleus starts to unwrap, and the edges of the cell themselves start to become less organized. So this is a dysplasia. And then finally you get uncontrolled growth. So you have a larger nucleus, disorganized edges, or edges of the cell are dysplasic. Basically, you can think it's not communicating well with its neighbor cells, and then growth. These are just a couple images. Normal. This is, hopefully you can see that this is going to be a malignant cancer. This is the cancer growth right here. You see that this is clearly going across this boundary here, right? So this is invading neighboring tissue. This shows you dysplasia, right? So these are the nuclei of these cells. They're quite large, and the tissue shape then changes, right? These are in this kind of nice bands. It's probably endothelial tissue, probably digestive. And then this is kind of all over the place here. So, squamous cells, skin. All right, so now you probably have heard of cancer stages. So I'm going to talk a little bit about the staging of cancer and how that's done. So there's 6 criteria in order to define cancer. And as we go through, I'll talk about all those criteria. But to have stage 1 cancer, you have the first 4 criteria. You have what we've talked about already, the first 2, division despite the proper signals, and division despite contrary signals. So that's important, right? Avoidance of apoptosis. This is programmed cell death, right? So cells are supposed to monitor their own DNA. When they see that there's an issue with their DNA, they are supposed to undergo a process where they destroy themselves. So if cells are not doing that, they're avoiding apoptosis. And then there's this thing called telomerase lengthening. I'll try to describe this briefly here. Maybe I'll put a video in about just simply telomerase lengthening. So DNA, let's say this is a strand of DNA. You know that for DNA, DNA basically has information, right? So these are codes that are going to give you different genes, okay? Right at the end, there's a bit of garbage DNA. That's this blue stuff. That's a telomere. And the reason it's there is that every single time the DNA is replicated, the telomere, this has to do with the way DNA is replicated, the telomere ends up being a little shorter and a little shorter and a little shorter. So after several hundred, sometimes anywhere, it depends on the length of the telomere and the mechanism, but after a certain number of replications, without lengthening that telomere, the cell has to stop replicating, or it's going to start cutting in to the DNA that codes for proteins, and that will kill the cell. So all our cells have the ability to lengthen the telomere, so that doesn't happen. But generally, that ability is turned off in cells that are not supposed to be replicating, and it only exists in stem cells and other cells that are supposed to be replicating. Cancerous cells have the ability to extend this telomere indefinitely, and when that happens, they can divide indefinitely. So then there's this, when we talk about absence of proper signals, this is going to matter a little bit when we get to the next slide. But these signals can be extracellular, so they can come out from outside of the cell. And those are often called NIBs, non-protein molecules. Okay. Why does that matter? The example I'm going to give you is breast cancer and Herceptin, or the HER2 membrane. So I'll try to draw this over here. Here's a cell, and that cell has a question, right? The question is, should I be dividing or not? Is it appropriate for me to replicate? And one way that that cell can answer that question is it can have surface proteins on it, right? That's not very accurate, but so let's call those 3 red things, okay? HER2 membrane proteins, okay? And those proteins are just sitting there, and they're waiting for these specific nibs, so these little molecules, to land on them. And when they do, it says to the cell, hey, yeah, you probably should divide. It's important to have breast growth or whatever the factor is. It looks like we need to divide. These are the growth factors called mitogens because they cause mitosis, right? So these growth factors cause mitogens. Well, what can happen with these breast cells is in breast cancer, what would happen if all of a sudden, instead of having 3 membrane proteins, you see that this one has 3 membrane proteins. Let's assume that this one is working correctly. Let's say you have 30 membrane proteins. So all of a sudden you're making 10 times the number of membrane proteins that are asking, should I be dividing? So even if everything else is the same, even if you have the same number of growth factors, the message to this cell is yes, you should be dividing, even though the message here might be no, you shouldn't be dividing because the growth factors haven't changed. So this cell is going to start dividing, and each time it divides, right, because its DNA is now saying make 30 of these protein receptors, all of its progeny are going to have too many protein receptors, and they're going to divide, right? And so this is one example of a way that you can get cancer. So Herceptin is a drug that's being used, and what Herceptin does is it binds to the outside of these receptors. And so it blocks the message because now the growth factors can't get to these receptors because the Herceptin's there. And so this is a mechanism of stopping cancer. So I'm going to go over lots of different mechanisms that we stop cancer with because there are as many mechanisms to stop cancer as there are types of cancer. And there are as many types of cancer as there are ways for a cell to misinterpret the message of division. All right, so now we're going to look at stage 2 cancer. Okay, so what's going on? So we have all of these first 4 stages, right? This is stage 1. The next stage, super important here, is angiogenesis. So this is the promoting of blood vessel formation. So this happens in all, I think I talk a little bit more about this. I talk a little bit more about this later on, but basically right here. This happens in all cells when cells become hypoxic, right? So, if you think about it this way, it's a super clever way to grow blood vessels. So remember this surface area volume issue. Imagine that you have like cells and they're growing here. This is an embryo, and it's getting bigger and bigger. And you have a blood vessel here, so growing along. Well, the more cells that start to grow, they're going to basically push these cells away from the blood vessel. When that happens, these cells, let's call this cell Susan is going to get sad, and she's going to be like, hey, I don't have any air. I need air, okay? She is going to release a chemical called VEGF. And that molecule is going to basically just float along, but eventually it's going to go on to that, I have an image of this. It's going to go onto that blood vessel, and it's going to cause a new blood vessel to grow there so that Susan becomes happy. Yay, happy Susan. And that's normally what happens. It's a very simple, direct, and effective way to make sure that every tissue in your body can have enough oxygen. This matters a lot when you get to cancer because, as you can see, what I just described is kind of normal tissue growth, right? That's supposed to happen. But the exact same thing would happen if you had a cancerous growth, right? And without the ability for cancers to do this thing, angiogenesis, genesis is the birth, right, or creation. This is the birth or creation of blood vessels. If cancer cells can't create blood vessels, then they are going to be self-limiting. They're going to die because they will not have any oxygen. So this is something that's going to matter a lot in stage 2 cancer, is they have to get around this. There's another drug here. So I'm going to, I think, might talk, there's another picture I'm going to show you with that. Sometimes I have a hard time, I usually have a hard time pronouncing some of these. Paclitaxel is another type of drug. So I'm trying to show you all these different cancer drugs so that you can, it's scary, but you can feel good. This prevents the formation of the cytoskeleton. So that's the architecture that holds the cell together. This is also what's used to make spindle fibers. So take a couple minutes and think about why the prevention of the formation of spindle fibers would matter if you're trying to prevent cancer. Right? Because spindle fibers are used to separate chromosomes in the stages of mitosis. So if you can't make spindle fibers, you're going to stop mitosis. So this is another drug that can be. I think we're going to skip the anti-CDK right now so I can go. This is now stage 2, stage 3. You're promoting blood vessel growth. And I want to actually get right here because this image explains how angiogenesis works. So here are some cells. They don't have enough oxygen, right? They have hypoxia, right? They're releasing VEGF. That VEGF goes to the endothelial cells of a really small artery. It's going to cause those cells, because they're listening, all these cells are listening too, right? It's going to cause these cells to change and basically form a new blood vessel. So this cell will start to grow, and actually as it moves, because you can imagine this is an empty space, there are actually cells here, right? As it moves, it comes in, macrophages come in and destroy these cells and kind of blast out the forest to build the highway, if you want. And these grow along and grow along and grow along, and then start to bring blood to the tumor. And this matters a lot because without this ability, without angiogenesis, tumors are basically limited to about 2 millimeters. So that's 2 millimeters in length. So that's very small, right? So if we could stop angiogenesis from happening with tumors, we would be in a really good place as far as fighting tumors. There are 2 drugs, Nexavar and Avastin. Both of them will basically attack VEGF and prevent it from binding here onto the arteries. Now that's fine until you cut yourself, or your liver needs to continue to grow, or you want to have hair that continues to grow, or anything like that. Then that's a problem, right? So it'd be good if we could have a more targeted approach to dealing with these cancers. And I'll talk, I think, a little bit about that generally. And at the end, I'll talk about kind of general and targeted approaches. I'm going to go back up. Yeah, I think I'll skip that and wait till the end. So you'll note that I skipped past a couple of these slides. I'll address those at the end. So the other thing that is true about cancer, it's gone beyond the envelope, and it's starting to invade outside of the tissue area. So I skipped through that kind of quickly to get to the angiogenesis. So this is an example of having it invade, basically move through other tissues. So when that happens, you're moving from 3, you're well into 3, you're in kind of the end of 3. It's going into other tissues. What tissue is it going to go into significantly? The lymph tissue. Why is that? Based on what we've talked about the lymph system, you should understand that if things are not where they're supposed to be, the place they're going to end up is the garbage dump, right? Which is the lymph system. So you're going to end up with basically cancer growing in your lymph nodes. That will happen. Or it could go into the blood system. Now, the reason this matters is the last stage of cancer staging is when you're getting to cancer, basically to stage 4, okay? And what this means is that the cancer can not only continue to divide, but it will break off from its body. So like this little dot right there is cancer. When it gets into the lymph system or it gets into the blood system, this cell will detach from the mass and will travel, and then it will re-adhere to a new location and start dividing. When that happens, cancer will become systemic, so you can get it anywhere because the cell masses are going to stop, go somewhere else, and be connected. So it's particularly problematic for it to be in the lymph or the blood system because that's our mechanism of moving things around. So at that point, they say the person has cancer all over their body. This is what's going on in that instance. Okay. So I'm going to go over just a couple. And the idea of these therapies is to give you a sense of how to address cancer. So you can have targeted therapies and general therapies, okay? So you can have radiation. Radiation interferes with DNA that is replicating. So when you have radiation treatment for cancer, that's what's going on, okay? Is that general or targeted? You should see that that's general, right? Because you have hair, digestion, skin, lots of different DNAs replicating, and it's supposed to be replicating, right? This is the reason that people who are under cancer treatment will have their hair fall out. They will have a difficult time digesting. Food will taste bad, because all of these cells that are supposed to be replicating are not going to be doing it. Again, chemotherapy, which will stop cell division and stop angiogenesis, okay? This is going to be general. This is, so I'm going to put a capital G there and then a lowercase g here. Obviously, angiogenesis is a more limited version of what our body needs to do than just general cell division, right? So chemotherapy, and chemotherapy does both, but Avastin, right? And the other drug whose name I forgot, right? This is kind of a better, more specific version, but it's still general, right? But then you can think of things like the Herceptin, right? Herceptin is really much more targeted, right? And that's going to be looking for cells that have too many HER2 proteins in them, and that's going to have an effect on those. You can get even more specific, and much of cancer therapy is looking at this. And this would be using these NIBs, so these inhibitors, either small or large inhibitors. And the thinking is simply, if you have a cell and that cell is cancerous, it's likely expressing a protein on the outside that's unique. Okay, so this is a blue squared protein. So that is to say, all of the other cells around it, all of the cells in your body, are not expressing that blue squared protein. So if you can find, or if you can train, so this is a green white blood cell, WBC. If you can train a white blood cell to recognize this specific protein that's on the outside, then that white blood cell would actually target and kill that cell that's part of your body that's cancerous. And so this is where we'd like to get. This would be kind of the gold standard, where we could train white blood cells to look for specific markers on cancer cells. So just to review, we have general cancer therapies. They're going to target any cell that's dividing. And then we have specific cancer therapies that are going to target cells that have particular profiles, proteins on the outside, or particular behaviors that are related to their division and or their tissue type. So maybe it's a pancreas cell or something like that. And then I wanted just to show you, to give you a sense of the types of cancers. CNS is central nervous system, right? So you can have those cancers, lymphomas. This is blood, right? This, by the way, these are the most amenable to cancer treatment because of this hematopoietic cell. We can pull out, assuming that it's not the stem cells that are cancerous, we can pull out stem cells, destroy all the other cells in the human body, and put the stem cells back in a human, their very own stem cells, and those can be taken care of and addressed fairly effectively. Epithelial, carcinoma, skin cancer, things like that, or your sunblock. Gonad cancer, so this would be germ cells, sarcomas, bone tumors. So these are all different types of tumors, and how specified they are. Leukemia, this is blood tumor as well. So lymphoma and leukemia, because this is a moving fluid, you're better off there. And then finally, I want you to take a look at these two cancers. These are two different types of, basically this is kidney cancer. This is two different images of kidney cancer. And the question is, which do you think is benign and which do you think is malignant? Hopefully you can see that there's a pretty well-defined and isolated edge there. This cancer has not grown out of its barrier, whereas this one has. It's spread out all over, much more difficult to address. All right. This last image we may go over in class. It's quite complex, but it shows you the role of cyclins. Cyclins are these proteins in the body that tell you when to perform mitosis. And if you did this like this, if you did G1, S, G2, M, different cyclins are going to go up or down in different areas. And one way that we found to be very helpful at addressing cancer is there are these things called kinases that help the cyclins move along, is to use these cyclin-dependent kinases and stop basically the buildup of these cyclins. When you stop the buildup of these cyclins, then basically you're doing the same thing as telling the cell, no, do not divide. So it kind of short circuits all of these DNA, everything in here, the DNA changes that happen that make cells cancerous and what's going on. So there's a ton of information here. You can look at it at your leisure. Okay.